Hello everyone, this is an introduction to the microbiology lab sessions, and also your bacteriology lab notes for today's lab. So your objectives for your microbiology lab sessions are to appreciate that the diagnostic cycle starts with you, the physician, who will order the appropriate patient sample, and then from there the appropriate diagnostics tools will be applied, which you will appreciate in our sessions. The various available methods, that is. And then finally, to also appreciate the lab report information. The different methods available for diagnosis include the non-cultural methods, which are not strange to any of you, such as the microscopy, the molecular tools, that is the PCRs and such. And then you have the culture methods, whether it's solid or liquid media. In our session, we will only focus on the solid media. Please take note of that. Well, in this particular session. And then the other method is the immunological methods, where, for example, you're detecting your antigen or antibody in the sample, the patient sample. So our particular focus in the microbiology sessions we will have is under bacteriology and parasitology, but you can appreciate that small note there that virology diagnostic tools most available are the antigen detection and PCR. So this session is focused on bacteriology. So our session is in two parts. Part A, which is the culture media, and Part B, which is antibiotic resistance. So Part A is culture media. You're all familiar with culture media and what it is. We had a session in your third year about this. But this time around, we just need you to just appreciate actual clinical samples on the culture media. And so we are trying to grow bacteria on agar, of course, and then identify very clear isolate colonies. Okay, and then we study them. We study their morphology, which is the colony morphology. And we use these colony morphologies to determine the identity of the bacterium. So I have a picture there of just the culture plate, just to jog your memory, because you did look at the culture plate in your third year, but this is a culture plate of a mixed growth, a mixed bacterial growth. And I wanted you to appreciate the separate colonies that are coming out, and you've seen them nicely clumped, identified as coming from one particular bacterium, okay, or numbered. But the idea is these are colonies, and the agars we have in the session today is not a mixed growth, it's a single bacterium growing there, but this is just to jog our memory about colonies